Hello, students. Let's wrap up what we've talked about so far about cell signaling, move on to cell feedback cycles, and then we'll be done. So exciting. And then we get to cover PMAT, prophase, metaphase, anaphase, telophase, or which are the full names of what we call the different parts of mitosis and cytokinesis. But before we do that, let's wrap up. So I wanted to wrap up our last topic by talking about an AP question. And so I'm going to read an AP question for you. If I figure out how to share my screen, I will. Let's see. Share now. Been trying to figure this out. Yeah, I know there's a way to do it, but I don't know how to do it, so I'm just going to read the question. So I noticed studying this that they asked about two things a lot. The first thing they like to give is talk about cell responses, and then have you talk about how that will relate to the structure of the transduction cycle. And the other one is to talk about mutations. So you really want to pay attention to know how can mutations affect the cell transduction cycle. So I noticed, for example, in 2018, I would encourage you to look at question 8 in 2018. It asks about acetylcholine receptor proteins that are found in the synapse between neurons and skeletal muscles. They said a researcher investigating two different types of the acetylcholine receptor proteins. There's two types, and they exposed them to different types of proteins. Type 1 is activated by both acetylcholine and nicotine, and type 2 is activated only by acetylcholine. So that's a typical, type of FRQ question. I would definitely encourage you to study that as you study the topic. Now, what's important here is, of course, many of these receptors are specific to one ligand. That's the whole idea of a ligand. It's a specific chemical. It could be a hormone. It could be something else, but specific to the active site of that protein. When it binds, it causes the conformational change. Now, this is a problem. This receptor can both get acetylcholine and nicotine. Obviously, that's a problem because the nicotine could be affecting the ability of acetylcholine to bind to that site. And so it's going to create physiological problems. So the first question said, describe this difference in structure and function between the two types. So you wanted to say that because one of them can bind to nicotine and one of them can't, they have to have a different binding site. So either that one has an extra binding site or the binding sites are different in shape. And that leads to it being activated by the type 1 being activated only by one and type 2 being activated by two different ones. And you can describe how that relates. The important thing here is that because they can both be activated by acetylcholine, there's no difference in their response. The difference is one type is more receptible to also binding to nicotine and the related problems that it might have. Then they said acetylcholine esterase is an enzyme that breaks down acetylcholine in a synapse. This makes sense. Remember, we said all of these processes have to have the ability to break down. So we don't want them to just keep running forever. That's the way that these processes are fine-tuned. They want us to describe the effects of inhibiting acetylcholine in muscle cells in type 2. Well, the important thing is, if we don't have this enzyme that breaks it down and inhibits, we know that will result in continued activation and in this case, the repeated opening of the sodium channel. So that's not what we want. We want something that we have enzymes to activate and enzymes to deactivate. And so if we have perpetual or continued activation without the inhibiting happening, that's going to, again, create problems. Now, the important thing, you don't have to explain, do a whole paragraph for the AP test. If it says describe, keep it short, couple of sentences, keep it to the point, make it easy for the graders to grade for you. And this is when, this is an example to tie in our last topic before the cell cycle. This is an example of negative feedback. Whenever we're inhibiting or have the ability to inhibit a signal, this is negative feedback. And this is used all throughout cell signaling. That we activate something, but we have inhibitors, another enzyme that come and inhibit. This is happens with our G proteins. If you remember the G proteins, the GTP it gets activated, and it causes a signaling cascade that can often have to do with phosphorylation of many enzymes. But the important thing is, eventually, that GTP is going to be broken by an enzyme and converted back to GDP, and that's going to stop the whole process when enough of the response is done. And so that's negative feedback. It doesn't just go on forever. We have the enzymes there to shut down the process when needed. So that's negative feedback. When can we have positive feedback? Well, I'll give a shout out to the excellent creators who are doing a great job in the AP classroom. Check out their videos. But the example they give is that plants, apples, for example, release this gas called ethylene gas, and that causes the nearby fruit to ripen, and so the whole orchard could start ripening as more and more gas is released. This type of using a gas as a hormone is very common in plants because the gas can diffuse easily through the cell membrane and cause the result. So rather than doing our traditional steroids like we see in humans, the use of gases as hormones is more common in plants. And then the other famous example of positive feedback that we always talk about in our biology class that you should remember is you should also think about childbirth, because the release of hormones is the speeding up of childbirth, which is very important for it to be a healthy childbirth. And so that amplified signal is what allows the childbirth to occur because as there's pressing of the child giving birth, then that actually releases more hormones, causing further dilation, and that's going to speed up the process, which is good because we want the birth to happen quickly and safely. So that's feedback cycles. Negative feedback is like a thermostat trying to keep things in control. Positive feedback is less common in our biological systems, but it can still occur. That's when a signal, that's when some sort of feedback amplifies the signal that's causing it. Another famous example is like a wildfire, because it's going to dry out the nearby brush, causing it to amplify or grow exponentially. Okay, that's it for today. So I hope that you now fully understand cell signaling. Definitely look at that practice question, 2018 FRQ 8, and other FRQs. A lot of them, again, talk about structure and function, and a lot of them talk about what happens if there's a mutation or some reason this process breaks down. You should be able to describe what will happen if that occurs. And that just comes from understanding each step and the function of each step in our signal transduction pathway. And then you should understand positive and negative feedback and the difference and how they're used in biological systems. I hope that helps. Have a great day.